Welcome everyone to our educational webinar, The Road to Functional Human Neuronal Circuits in Vitro. My name is Sam Lloyd-Burton. I'm a Senior Product Marketing Manager at Stem Cell Technologies, and I'll be your moderator for this webinar. If you'd like to ask questions during the presentation, please use the Q&A panel or chat box on the right, and we'll address as many questions as possible at the end of the presentation. We're delighted to have Dr. Cedric Bardy as our guest speaker today. Dr. Bardy obtained a PhD in medicine from the University of Sydney in Australia and pursued his postdoctoral training in neuroscience at the Pasteur Institute in Paris and the Salk Institute in California. Dr. Bardy is an expert in human neural stem cell models and electrophysiology. His research efforts are focused on developing new stem cell biotechnologies and strategies to understand the effect of neurological disorders on brain cells. A few years ago, Dr. Bardy set out to identify which properties in traditional basal culture media could be altered to better support in vitro neuronal activity. His solution resulted in the invention of a new neuronal basal medium, BrainPhys, published in the Proceedings of the National Academy of Sciences. Dr. Bardy is here with us today to talk about electrophysiological measurements of human neural cultures and the advantages of neuronal media adapted for neurophysiological activity. Let me now hand you over to Dr. Bardy to begin the presentation.
So I wanted to share this moment with you and tell you a little bit about this progress we made on that road towards functional human neural circuits in vitro. And obviously this is a very large topic and there's many things to cover. So today I hope you won't mind if I try to focus mostly on how and why we developed this new neural medium called BrainPhys that I'm sure most of you have in the audience have heard about already. So I'm also assuming that most of you in the audience are very familiar with hPSC technologies. And it's still amazing to me that starting from the Nobel Prize discovery of Yamanaka, we can now use 4 factors to reprogram skin cells, so any kind of somatic cells, in fact, to stem cells called induced pluripotent stem cells, or iPS cells. Now, the beauty of this technology is that you can then use a first set of differentiating factors and turn these stem cells not in skin cells anymore, but any kind of lineage that you might be interested in, from mesoderm, endoderm to ectoderm. And from there, one application is to use this lineage for cell therapy in vivo, like cell transplantation. And another application that I am personally very interested in is to use this lineage to model human tissue in vitro and also model human disease in vitro. So being interested in the brain, you can start with ectoderm lineage and use a second set of differentiating factors that can be used to pre-pattern neural progenitors into any kind of brain cells that you're interested in, from glutamatergic neurons, GABAergic, dopaminergic neurons, serotonergic, motor neurons, and even other type of brain cells like astrocytes, oligodendrocytes, or microglia. Now, the idea is that you can take all these cells together in a dish or in a plate and let them mature for some time, so then try to guide this maturation and obtain what one may call a mini human brain in vitro. Now, the good thing is you can repeat the same protocol not only from patients but also from healthy subjects, and then end up with a live human neural tissue from the patients or from the controls. And once you identify differences between those two tissues, you can use directly pharmacology in vitro to compensate or revert those differences. And of course, once you have an interesting compound in vitro, then you can use it and test it directly back in the clinics on the patients. So my main objectives in the lab is really to use this model for disease modeling. But today I'm not going to talk too much about the disease part and focus mostly on trying to make sure that we have the right conditions to make this model as useful as possible. Something also I'm very fascinated by, I've been fascinated by that for decades since I really started science, to be honest, is electrochemical activity in the brain. I love this because everything we care about as a human being, the way we feel, the way we behave, the way we take decisions, the way we communicate with each other or even learn a new language is, and even the way we conscious about all these things, is somehow directly connected with electrical activity in your brain, namely firing of action potential and synaptic communication. What's interesting too is that the same electrical and synaptic activity is also very important at the cellular level, and it's been shown many times in the neurodevelopment that electrical activity can influence the cell migration, the maturation of the cell themselves, or the formation of dendrites and synaptic connection. Even the synaptic strength is strongly influenced by activity through LTP, and even though the survival of the cells in the brain can be influenced by activity. So given the context, of how important electrochemical activity in the brain is, it seems obvious, but it's very important that we pay attention to that also when we model human brain neural tissue in vitro. So we can also learn from experiment done on animal in vivo, and these are two examples of cells. On the left, you have this is a pretty heroic experiment where researchers have patch clamped single neuron in the right primary visual cortex, and so on the left you can see this trace. Basically, it's a patch clamp neuron, so in a live animal, and you can depolarize the membrane potential with a step of current and evoke a train of action potential. So this is pretty artificial, and you can do the same thing again in vivo, and instead of depolarizing artificially with a patch pipette electrode, you can present a visual stimuli to the animal. And in this case, we're in the primary visual cortex, so the cells are well known to respond to gratings or bars, so you can move a bar of a specific orientation, direction, and velocity, and evoke a similar train of action potential with this more natural stimuli. Can do the same thing in the other sensory part of the brain, like the olfactory bulb in mouse, and this is, I believe, mitral cells recorded, and you can evoke the same thing, the train of action potential artificially, or more naturally with others. In this case, it's quite cool. I can see the rhythm of the respiration, and so when there's no odors presented to the animal, like the firing or the membrane potential at rest is kind of flat, and then when you start presenting the odor to the animal, then you start having a burst of firing in synchrony with the respiration. So this is just to give you a very quick idea of what electrical activity could be looking like in the brain in vivo. Now, of course, it's very difficult to do the same experiments in human. The closest we've been to is from maybe experiments done by others on human brain slices from fetuses or biopsy from epileptic patient or cancer patient. And of course, in this case, you cannot present live natural stimuli, but you can do the same thing and depolarize the patch clamp cells with step of depolarization and have a very similar train of action potential that you can see in vivo. So the interesting thing here is when you do these experiments in vitro or ex vivo, you need a solution. We need to have the cells in a medium that support the electrophysiological activity. And for decades, electrophysiologists have used ACSF, so which stands for artificial cerebrospinal fluid. And this is a very simple solution comprised of inorganic salts, energy substrates like glucose and phosphate buffer. And this is great for electrophysiology, and you can see it will be pretty much mimicking the environment that is in the brain in terms of supporting electrophysiology. Now, the problem is like this ACSF cannot be used with tissue culture because there's not enough compounds in there that support the cell survival for more than a day or so. So the question we asked is, do we have actually conditions in tissue culture in vitro that really support the basic functions of human neurons, so firing action potential and synaptic activity? To answer this question, we did these experiments where we started, so we differentiated the neuronal culture from iPS cells with protocol that we had already established, and we went in and patch clamped those cells in a CSF. So for those who are not electrophysiologists in the audience, patch clamping is an electrophysiological method to look at as much details in terms of function as you can go, you know, from action potential, synaptic activity, etc. The way it works is you have a patch glass pipette that is connected with an electrical amplifier, and you can access the differences in membrane potential of the cell and currents of ions going through that cell. So you can record the activity of a single neuron. So in this case, you can see a spontaneous firing of action potential over time. So each spike going up was an action potential or a train of action potential. You can also look synaptic activity, both excitatory, glutamate, AMPA-mediated activity. So each spike going down one eye, it's a release of neurotransmitter onto the neurons that you are patching. And or inhibitory activity like GABAergic, the kinetics of the currents are slightly different and slightly different properties. You can complement electrophysiology or patch clamping with other techniques like calcium imaging to have a look not just at much details into one cell, but more like a population. So you can see in this movie, each cell firing, flashing with different color would be when there's a massive amount of calcium increase in the cells related to firing of action potential. So this is great, but the dirty secret, like my mentor Rusty Gage likes to call it, is that we grow the cells in a medium, but then when we test it for the physiology, we use a different medium, which is, in this case, ACSF. And so what we did is like, well, what about if we test the same cells in the medium that we use in the incubator to grow them? And when we do that, what we found it was a dramatic effect on the electrical activity. So in this example, we use DMEMF12, which is a traditional method that most people use for neuronal culture. And what you can see first on action potential, spontaneous firing, you have a decrease of the resting membrane potential that lead to a complete silencing of action potential firing. Same thing happened with synaptic activity, both glutamate and GABAergic activity, a silencing of the synaptic activity. And when you look at a more population level with the calcium imaging, you can see that the only difference here is really the perfusion in the bath of the medium and the same field of view, same everything. And you can see that the culture here is much less active than what it was when we were in ACSF. You still see a few cells that are active, but much less. Now, it's no artifact because you can perfuse back the original solution, in this case ACSF, and recover mostly the response if you wait long enough. Now, DMEM is one medium that is largely used for tissue culture. Another medium that is used also for tissue culture of neurons is neurobasal. So we did the same experiments with neurobasal. We started in a CSF and look at synaptic and activity and action potential. Then we perfused a neurobasal. And what you can see is also a dramatic impairment of electrical activity by the medium. The composition of neurobasal and DMEM is very different, so of course, the parameters are slightly different. But what you can see, so if you start with glutamatergic activity, it is very strongly reduced, even though you can still see a few spontaneous events. GABAergic activity is completely silenced, like in the MEM. And action potential firing, so the resting potential doesn't change this time, but you can see that the spike are not as great as they would be in a CSF, and that's supported by weaker sodium and potassium voltage-gated channels. Now again, it's no artifact because you can recover the response by perfusing back the ACSF. Again, if you wait long enough. So what I'm telling you is that we have great basal medium, that support survival and even differentiation of neurons in vitro, like DMEM and basal. But they are absolutely terrible for fundamental function of neurons, like firing action potential and synaptic activity. And on the other hand, we have a CSF, which is terrible for cell survival and differentiation, of course. And but it's great for fundamental function and activity. So what we decided to do is maybe we need something in between, something that can take the best of all these medium. And that's what we decided to try to design BrainPhys. And so we wanted something that can support the survival and differentiation at least as well as DMM and basal. And we wanted also the same medium to be able to support fundamental function in vitro, because we believe it's important not only to generate neurons that can fire action potential, but to have them, firing and this action potential and communicating through synapses while they're in incubator, while you do the experiments, not just when you test it for electrophysiology with ACSF. While we were at it, we also thought we'll try to improve a few other things, like the glucose level in this medium is very high, something like 2 times or up to 10 times higher than the glucose level that is reported in hyperglycemic patients. So I don't know exactly what influence it has. It might not even have any influence on the activity, but overall I think more changing it to more physiological level might be an advantage for disease modeling later on. So let me take you first to a bunch of results showing you that ACSF and BrainPhys support optimally the electrical activity and to the same level. So this is an example of action potential. So you can now do the same experiments I showed you before. You record in ACSF, this is spontaneous bursting of action potential or more sustained kind of firing. And you can record the same cell in ACSF or BrainPhys and have very similar response. You can also evoke action potential with a step of current and find no difference between the two medium. You can also support this with looking at sodium and potassium voltage-gated currents. So, of course, each cell might be slightly variable in the amount of currents you express, but when you switch from one medium to the other while you're recording, there was no difference whatsoever. So then the other thing you can do is you can now use optogenetics, so you can express channel rhodopsin, for example, in a human neuron. And in this experiment here at the bottom, we had the cell in current configuration. We injected no currents. The only things that changes is the solution that is perfused in the bath. And what we're recording can flash a brief flash of a blue light on this neuron expressing channel rhodopsin, and the channel rhodopsin will open, have a little entrance of sodium in the cell that will then trigger natural, action potential mediated by sodium and potassium voltage-gated channel. You can, so each time you flash the light, you can evoke a nice and clear action potential. And the only thing that we change here is not the solution that is perfused, and you can see that you can have exactly the same response in BrainPhys. Now, if you were doing the same experiment, the same optogenetic experiments, in this case in DMEM F12, so you look at this example here, again, same thing, flash the light, get beautiful action potential evoked by the light, and when you, the only thing we change here is the medium, in this case DMEM F12, and same thing as earlier, we get like depolarization and silencing of the spike. You can do that in basal as well, and so in this case we started in BrainPhys. You get beautiful evoked action potential. You flash a light, and you do have some response, but they're not very great, and I don't know, we can even call that a spike. But you do have some light evoked response, but it's much poorer compared to the action potential that we could record in BrainPhys. And this is different trials that we did, so it was pretty consistent. Again, you can support that with recording in the same condition, voltage-gated sodium and potassium currents, and quantify all this and find that the currents are much reduced in neurobasal compared to BrainPhys. So overall, I'm telling you BrainPhys basal support optimally action potential firing. Now, what about synaptic activity? So we did the same experiments and looked at synaptic activity, both AMPA and GABA. First of all, we find no differences in the synaptic activity when we recorded it in ACSF or in BrainPhys. In contrast, when we compared the synaptic activity when we recorded in BrainPhys or in ACSF compared to neurobasal, the neurobasal was much reduced. You can see the summary of all the experiments here. It was dramatically reduced in all of the experiments. Same thing for a GABA GK event, and in DMEM was totally reduced. You can add a little bit to that and just look at the calcium activity either in ACSF or in BrainPhys of the same field of view, and we find no obvious differences between the activity in the two conditions. Now, BrainPhys, neurobasal, DMEM, it's all basal medium. So it's the basic nutrients that the cells and salt that the cell needs to survive. But you also need to add supplements, and so we decided to test also the supplements that people typically add to tissue culture and see how these affect electrical activity. So the first one we tested was serum. Serum have been used for quite a long time now, a little bit less maybe in neuron culture, but still some people use it. And it was pretty clear that serum was very dramatically impairing electrical activity, a little bit like DMEM, so that's quite interesting to think about what's in there. But basically, if you look at spontaneous action potential firing, then in ACSF in this case, and you can now add 10% serum to the same ACSF, and you see a depolarization of the resting membrane potential and the silencing of the firing. We also repeated these experiments in what might be called more long-term experiments on NES. So in these ones, we used cells from cellular dynamics, so supposedly hPSC-derived human neurons that were mature, and so we can thaw them. And so the day zero here on this MEA is multi-electrode arrays is mature neurons. We start from mature neurons, and you need a few days for them to wake up and thaw in the medium. And if you do that in BrainPhys plus serum-free supplements, you see a very, very nice activity. In fact, a lot of active electrode in the MEAs, and the frequency is quite decent. But if you add serum to BrainPhys, then the activity is completely silent. So it's very important to keep that in mind. So, of course, if you add serum to neurobasal or DMEM, it's also reduced dramatically the activity. So serum impairs neurophysiological activity, so unless you really need it, you should pay attention to that. Now, there's alternative. You can add serum-free supplements like these ones here on this list, and we tested them in conditions, and we used BrainPhys with supplements or without supplements, and we find no obvious differences in the electrical activity where we did these experiments. So overall, what I've told you so far is that first, classical basal medium and serum impair activity. Neurophysiological activity of mature human neurons is optimal in BrainPhys plus serum-free supplements. Now the question is, can we also use BrainPhys for development and maturation of human neurons in vitro? So to answer that, what we did now is, instead of using BrainPhys acutely, we start with fibroblasts, and we program them into iPS. We start directly from embryonic stem cells and convert them into neural progenitors. Then we seeded the neural progenitors for 24 hours in a plate, and then gradually switch the medium that we feed them with BrainPhys plus serum-free supplements. So within a week or so, the medium will be replaced by a BrainPhys plus supplements. And we waited more than a month in those conditions in this experiment, and we then analyze the neurons. So we stain them, we find that after a month, in this case, the cells made a beautiful neural network that can be identified with MAP2 dendritic staining here, or TERJ1. We could, differentiate different kind of neurons from dopaminergic, GABAergic, obviously glutamatergic as well. And we also could stain for synapses, synaptic contact with synapsin and MAP2 overlap. And you can zoom in and you can guess here maybe that's an axon, and this is a synapsin at the dendritic contact. Then we wanted to see if these contacts were functional, so now you can do the patch clamping in the same condition that I use for maturing them. So in this case, BrainPhys plus serum-free supplements. So all this patch clamping was done in this medium. And you can evoke, nice action potential, find, like optogenetic driven action potential, sodium current, potassium current, and beautiful synaptic activity, both AMPA and GABAergic, and also at the population level, you can see a nice calcium activity. So that's quite encouraging, and we were very pleased, and this is really what we wanted to do. We wanted a medium that support both survivor differentiation and also optimally support electrical activity. So from there, we were. So I told you earlier that electrical activity is also important in vivo for neuronal development. So it was obvious to ask, well, do actually, growing the cells in BrainPhys may actually improve the neural development. So we thought we should try to ask this question. And so to do it, we did these experiments where so we started from human neural progenitor cells, and then we culture them so we seed them in a plate, in a 24-hour plate, in this case on glass cover slips with no astrocytes. And we seed the whole plate with this neural progenitor, the same density, same everything. And then half the plate had BrainPhys plus supplements, and the other half had DME-MF12 plus the same supplement. And we then did a blind, so we let them grow for between 3 to 10 weeks. Then we did a blind analysis in ACSF after long-term exposure to this different medium. So it's important to understand that, of course, we did the patch clamping in this case in ACSF, even though we grow the cells in different medium, just to be biased, because if we do the patch clamping in the MF12, I told you the activity would be impaired. Then we look at the data and put it together, and what we find was pretty cool, actually. We found that the group that were grown in or fed with BrainPhys medium plus serum-free supplements had, so the number of cells we patched, there were a much larger percentage of cells with active spontaneous AMPA-mediated events even after 3 weeks, and in comparison to the cells that were fed with DME-MF12 plus these supplements. And this is also seen in the frequency of the synaptic inputs, and these experiments were repeated also by stem cell technologies or their collaborators that were blind completely to the medium they were tested, and they seem to find very similar results after culturing the hPSC-derived neurons for more than or about 10 weeks each. Same story pretty much with GABAergic as inhibitory synapses. So we find a little bit more number of neurons with active GABAergic synapses in the BrainPhys group overall, and the frequency was also higher. So everything I've shown you, and so here I'm talking about synaptic and silencing in BrainPhys, and the reason I'm using that word and silencing is when we actually try to stain for synaptic puncta on the neurons, we didn't find a huge difference between the cells that are grown in BrainPhys or in the MF12. So that suggests to me that the cells are able to form synapses, but because of the medium, they might not be as active as they should be. And by releasing the spontaneous activity in BrainPhys, then you're probably going to trigger some molecular events, but it's going to unsilence these synapses. So everything I've shown you so far is on human neurons derived from ES or iPS cells, and a lot of people in the field are also interested in direct neuronal conversion from fibroblasts directly to neuron, skipping completely the iPS cell or stem cell stage. And so we thought it was to try BrainPhys in this kind of experiments. So that's what we did. We tried it both, using it in the direct eye neuron conversion directly from human fibroblasts to eye neurons, and it seems to work fairly well. And we also obviously try to use it for eye neurons maturation. Now, for the conversion, when we quantified the number of neurons over the total number of cells, we didn't see that there were more neurons in the BrainPhys group compared to the. In this case, it was a mixture of DMEM and neurobasal plus the same supplements. So we didn't find, dramatically advantage of growing directly differentiating, converting the cells in BrainPhys in terms of number of neurons. Nevertheless, what we found is that if you let them in maturing in BrainPhys for long enough, then we looked at ARC protein. ARC is interesting. It's a gene that has been reported many times to be related to electrical activity and absolutely critical for learning and memory, and also for this unsilencing of synapses I was talking about earlier. And in these experiments, we looked at 3 different cell lines from 3 different healthy subjects, and in all of them, there was a dramatic or significant increase in the intensity of ARC protein expression in the neurons we analyzed. So obviously, BrainPhys was designed for human neural culture, but we know that the activity in the animal model, like the mouse, is very similar, at least for the basic, action potential and synaptic firing in animals. So we wondered if that could be used also for let's say rat primary culture in this case. And so we did these experiments where we isolated hippocampal rat E18 cells and replated them in BrainPhys for about 2 weeks or a little bit more, and let them mature and grow back, axons, and then right after dissociation, and then analyze the electrophysiology of these cells or staining of these cells again in the same solution in BrainPhys plus SM1. And we find beautiful electrically active neurons and obviously a lot of synaptic puncta stained by synapsin and MAP2. I believe stem cell technology have shared with me that they have also repeated these experiments, and they went a little bit further in that case where they compared the synaptic activity in BrainPhys with neurobasal culture after 2 weeks, and they seem to find that they find better or higher frequency of synaptic inputs in the BrainPhys culture, both mediated by AMPA or GABAergic receptors or synapses. So overall, I believe that BrainPhys can be used with a very different kind of human brain cells, and we've tested a few. Obviously, it can be used with glutamatergic and GABAergic neurons, but also other type of neurons like serotonergic or motor neurons maybe. And so we've tested that, not as extensively as what I've shown you or in everything, but we seem to have some success. Some people seem to have reported that it works specifically well for dopaminergic neurons, so there might be slight differences in the type of cells that you're using and the benefits you get from BrainPhys, but overall, I like to believe that the brain has the same, pretty much the same environment for most type of neurons. And same thing for astrocytes. It's definitely astrocytes are very happy in BrainPhys, and I don't understand necessarily myself why we shouldn't need a astrocytic medium versus a neuronal medium, but this is another question. So The take-home message here today are first, classic basal medium and serum impair fundamental neurophysiological activity, and that might be important for our experiments and trying to model human tissue as physiological as possible. On the other hand, I hope I convinced you that BrainPhys can support optimal neurophysiological activity of mature neurons. It can also be used for either short-term experiments, or if you really, really like the protocol you have and you believe there's no reason to change it, you can also maybe try to keep that protocol and obtain your mature human neurons and maybe switch to BrainPhys for short-term just at the end of the experiment. But we also show that we can also use it for long-term culture, at least in weaver conditions. Overall, we find that long-term exposure of BrainPhys and silencing functional synapses, which can be important for many people. BrainPhys can be used for human iPSCs or hPSC-derived neurons, or also for directly induced neuronal culture, and also for different species, not human only, but also rodent neurons. So, clearly we designed BrainPhys for cell, for experiments as scientists, and this is not a magic medium. We're happy to share the recipe. It's published, and we try to design it in a way that will make our experiments easier. For example, so of course, a lot of it was tried to make it as physiological as possible and mimic the environment in the brain and supporting electrophysiology. For example, we adjusted the calcium chloride to physiological level to 1.1 millimolar. Now there are some reports saying that higher calcium chloride concentration might be helping in synchronizing neurocircuit activity. And the good thing about the way we design BrainPhys is that it's pretty easy to add stuff to it. You can add a little bit of calcium if you want, if that helps you to with your experiment. So we hope it's as versatile as possible, and of course this is a basal medium, and you can still combine it with whatever supplements you want. So to conclude, I think the idea really is to try to improve our condition to make them more physiological, and the hope is that by doing so we will improve down the line translational success, because we're really in that game of using iPSCs and human neurons to get a model that is as realistic and close as possible as the human patients. So this is maybe one step further on that road, if you want. I'd like to thank now all the people that have significantly contributed to that work. It was really a teamwork, and it's wonderful to work with everyone. I'll start with Rusty Gage, my mentor. Is obviously a fantastic and amazing mentor. I'm very grateful to have been having this support for the last several years now. I'd like to thank also the electrophysiologists in the team, who've been working with me and helping me with a lot of patching, not only this BrainPhys solution, but many other versions that we had before. So Cynthia, Ruben, and Mark has been tremendous in patch camping. Mark has been helping with the calcium imaging experiments. I had a lot of help also with tissue culture from TJ and Leah, Mariko. Cynthia has been helping with the electrophysiology. I think I forgot her. Also, a lot of people have been helping me, like great help from Josh and Anne Bang on the AMEs, the multi-electrode arrays that they did. Jerome and Lena did all the experiments with the induced neurons, which was great. And Susan has been very helpful to giving me a lot of tricks to prepare this medium and try to modify them, and Mariko has been helping with all the immunohistochemistry. With that, I'd like to thank you all for listening, and I think it's all live, so it's time for questions, and I'm happy to try to answer as much as I can, and if I can answer your question today, please feel free to contact me. I'll be more than happy to try to help.
Okay, thank you very much, Cedric. That was a wonderful presentation. So that concludes the presentation segment of the webinar. We will now begin our Q&A session by addressing the questions that were submitted during the presentation. I also have with me here Dr. Vivian Lee, who's Senior Scientist and Neural Team Lead at Stem Cell Technologies. So if you do have any questions for either Cedric or for Vivian, please use the chat box on the right of your screens. Now I'll start off with some of the questions that were already submitted during the presentation. Okay, Cedric, could you comment on for how long you've been able to keep neurons healthy in BrainPhys?
Yes, sure. So usually we do our experiments for about 4 weeks to 5 weeks, but we tested them all up to more than 5 months, and then I did some patch clamping in there, and it was still very, very healthy and very, very beautiful. Now the trick is like any other medium, I believe, or tissue culture, the challenge when you keep them for too long is not always that they stay attached at the bottom of the dish, especially for myself where I use glass cover slip bottom for the electrophysiology, and I try to avoid using an extra layer of astrocytes. So I think the limitation here is not so much the medium, but the physical condition, if you want.
Okay, thank you. So could you comment on how many days do the cells need to be maintained in BrainPhys before you do the electrophysiological recordings?
Well, I don't think there's a perfect answer to that. I can tell you how long I try to keep them, and usually, again, I try it 4 weeks or so. But I think the time in vitro is really going to depend on your protocol and what you aim to obtain. Like the level of maturity. Of course, if you wait longer, might be greater. And so I don't think there is a clear number here what you should do or what you shouldn't do. But we try to do plan experiments around 4 to 5 weeks. And that makes them fairly practical now. I don't know if it's as mature as it should be.
Okay, thank you. Do you or any of your collaborators know whether BrainPhys has been used for a cerebral organoid model or a mini brain model?
No, we started that, but I don't have any really good insight yet. But I think it's definitely great. Like it's the same idea. Trying to make it more realistic. So yeah, this is definitely an interesting question, and unfortunately, I haven't done yet too many experiments with that. But I'm looking forward to it.
Great. We've had a couple of technical questions about the electrophysiology. Can you let us know how do you maintain the oxygen concentration at a physiological level while you're doing recording?
Yes, so while we're doing recording, we perfuse or bubble the solution with carbogen, which is I think it's 95% oxygen, 5% carbon dioxide, so while we're doing the patch clamping recording, that's what we're using. We just bubble the solution. Now, if you I don't know if the person who asked this question, if they try to bubble the solution with supplements and growth factors, then you'll see that it's going to start being foamy. So when we did these experiments with the supplements, in that case, I left the medium for some time in the incubator and just ventilating it, like the way that the tissue culture plate are, to avoid the foamy, situation.
Great. So we've had a few astrocyte-themed questions. So I'll start those by saying, traditionally iPS-derived neurons have been co-cultured with astrocytes in order to achieve better electrophysiological maturation and spontaneous activity. Do you find that co-culturing with astrocytes still makes a significant improvement even when you're using BrainPhys?
To be honest, I didn't compare extensively the two conditions. I do believe that having the right astrocytes in it might help at several. So first of all, the condition I'm using, and usually when we start with neuroprogenitors, you end up with a mixture So from this neuroprogenitor, you end up with a mixture of glial cells and neurons. So we do have astrocytes in the culture. Now, I am interested in this model for disease modeling, and I'm always a little bit reluctant to add astrocyte from another source because I want to, have the cells. I want to be sure that I have the cells I'm looking at are exclusively from the patients I'm interested in, and avoid some sort of contamination. But overall, yes, I think like astrocyte might be adding an extra layer of astrocyte in some experiments might be a good thing. It can help with the cells detaching a little bit less. But it's a trade-off, like if you're really confident that you're going to get any contamination from the external source of astrocyte, then use them. It might be better. There's many people showing that it's better, so there's no reason to believe that it's not.
Thank you. And if you're generating astrocytes from pluripotent stem cells, how long does it take before you get, functionally or morphologically mature astrocytes?
Oh, I don't know that. I've really focused on the neurons, so I don't know. I know that the astrocytes I have in my culture, after 4 weeks, they will stand for GFAP, they will stand for S100 beta. Now to call them mature, I don't know if I can go there, and I cannot answer this question. Sorry.
That's okay. So you showed a little bit of multi-electrode array data in your presentation, but we had a few questions about that. So could you comment on whether people have compared the activity of neurons cultured in BrainPhys using multi-electrode arrays and compared that to other media?
Yeah, so our collaborators, and some of it is published in the paper. I don't think I've talked about it today, but you can check it out in the PNAS paper. We tried it with serodynamic cells, and we found that the activity in BrainPhys was definitely much better for at least 2 weeks, if I remember well, in BrainPhys compared to neurobasal with the same supplements. And the MEM was in the experiments was a little bit funnier. It kind of went up and down, so I think it's also telling me that when you feed the cells with the MEM, especially if you have astrocytes in there, they might take some time to get rid of all the active neurotransmitter like glutamate, for example, in this medium, and so you might have less consistency over time, just so they can take it up. So the activity in the MEM was going up and down, where it was a lot more stable in BrainPhys. Now we've been trying to repeat a little bit more these experiments in different cell lines, and I've got very similar results so far. But I've seen also it might depend on the cells and the timing and everything. So some cells seems to be more sensitive overall in the activity than others in to BrainPhys. So maybe some cells expressing more GABAergic receptors than others, and things like can influence the level of activity and the detrimental effect of the other basal medium.
Thank you. So just amalgamating a few very similar questions together here. So are you able to say that neurons mature faster in BrainPhys?
Yeah, so I'm always very careful when I use the word maturity because it's a very loose term. But what I'm able to say is what I got from the experiments I showed you today. Like when we compare them in terms of synaptic activity, we had like higher number of cells with a functional and mature, if you want, synaptic activity.
Thank you very much.
And so sorry.
Sorry, carry on,
Yeah.
Cedric.
If I can add something, it's important for me to say that I did not design this medium to improve the maturity. I designed this medium to improve electrophysiological activity. Now, the fact that it improves the synaptic activity and the maturity, probably, it's a bonus for me. It's great. But I didn't try. I think to improve maturation, there's many more things that can be done, especially working on the supplements, adding the right supplements, and maybe organoids or 3D culture that might help also. But they can be used in combination with BrainPhys.
Great, thank you. That is a question we do get quite frequently at Stem Cell as well. So we have a few people who are working with mouse primary neurons, and they're asking about whether they should be isolating the cells and plating the cells immediately in BrainPhys, or whether they should be waiting a few days and then transferring to BrainPhys.
Yeah, so I think some for right primary neurons, maybe Carmen or you guys at Stem Cell Technology have more insight than I do, but my understanding is ideally it's better to plate them, to not plate them in BrainPhys immediately. I think you guys are using your basal, I believe, for the first 24 hours, and then to switch gradually to BrainPhys. I don't know exactly why is that. My best guess is that the fact that the cells are more active in BrainPhys when you start manipulating them, like switching them from one plate to the other might be not so great if the metabolism is quite high. So having them in a medium that is a bit less active might be better to transfer them, and then you can switch to BrainPhys. But you might want to add something to that. I don't know.
Yeah, I can add. So from the data generated at Stem Cell, we focused significantly on primary neurons and found that, although you can plate directly into BrainPhys, we get improved survival and improved function if you initially plate in your normal basal medium and then transfer into BrainPhys from around day 5. And the reason for that is exactly what you were saying, Cedric. I believe that the requirements for the cells during those initial few days are quite different from later on, where you're trying to just encourage survival and sticking down initially, and then later on, once the cultures are happy and on the dish, then transition to BrainPhys. And certainly in our data, we get a best of both worlds situation with that protocol. Okay, so time for just a couple more questions here. So I have one question asking about the membrane resistance, whether you see an improvement in membrane resistance in BrainPhys compared to other media.
Gosh, I don't remember exactly. It will be in the supplemental data of the paper. I believe we did see a significant increase in membrane resistance when we compare the cells with growing in BrainPhys or in DMEM for 3 weeks. I believe it was significant, yes, but you can check the numbers in the supplemental figure of the paper.
Okay, just trying to, we've got a lot of questions, so I'm just trying to pick out the key ones here that have come up a few times. Okay, so can you comment, a lot of them are about different applications, which, I guess you might know from collaborators, or, we have some collaborators. But there's a question about co-culture of hPSC-derived skeletal muscle with motor neurons. Are you aware of anybody having tried that, and whether they're able to get functional synapses?
That's a great question. I don't know. I know people who are trying that. I know people who have used motor neurons, BrainPhys on motor neurons, and I've actually in a collaboration I patched some of these neurons, and they were beautiful, beautiful activity after I think it was a couple of weeks in BrainPhys, or maybe 3. And but I haven't seen the co-culture with muscle cells and motor neurons. I'm sure people are working on that. I'm not working on that myself.
And other basic media, so DMMF12 and neurobasal, altering protein or channel expression or localization, or altering channel activity, or both? Do you have a sense for what's actually happening there?
Yeah, so so I'm not sure I get exactly the question, but clearly there is so the different aspect in neurobasal and the MEM. The MEM, I think the glutamate in there is clearly saturating the channel, the glutamatergic channel, and therefore depolarizing the membrane potential, through sodium entrance in the cell. In neurobasal, they remove glutamate, but they also reduce strongly the sodium chloride, and sodium chloride is absolutely typical for action potential firing, so that that's not working so great for in that sense. And both medium had additional active amino acids directly binding to GABAergic receptors or glycinergic receptors on the cells that have trigger chloride entrance in the cells that also affect dramatically the membrane potential and current activity.
Okay, thank you much. Thank you very much. Sorry, go ahead.
At the protein level, I think there's still a lot of things to look at. To be honest, I'm not that interested to continue comparing, very carefully the effect of neurobasal and the MEM or BrainPhys, because from a scientific point of view, it's so. I was interested to do it to show that okay, it works and it's like that. From a scientific point of view, there's so many differences in the three medium that even if I find a protein that is increased in one or the other, mechanistically it's going to be hard to tease out what exactly is the reason of that. So I think if you really wanted to address this question, you need to tackle one component at a time in this medium. So use one medium, whichever one you want, let's say BrainPhys, and add let's say calcium, or add one thing at a time to understand the mechanism of changing this on protein level and channel expression and things like that.
Okay, absolutely. It's a very complicated question to try to tease apart. Okay, that's all that we have time for today. I'd like to thank everyone for attending, and I'd especially like to thank Cedric for his time and fantastic presentation. A recording of this session will be available on our website soon for your viewing convenience, and we'll send out a link to this to all registrants as soon as possible. Thank you all again, and goodbye. Have a fantastic evening or day wherever you are in the world.